So we skipped 8.5 here, and most of these we were just touching on ideas. And there's also a 8.11 we're skipping. Basically like 8.7, I've got one slide. So not a lot here. I want the slide. Okay, so horizontal gene transfer. That's how bacteria share DNA, right? Because the only ways they can get new genes is through mutation or horizontal gene transfer. All right. And if you've ever transformed bacteria in another class, this is what you were doing. You were doing horizontal gene transfer. Once we give those bacteria new DNA, they're known as a recombinant. So the word recombinant just means they have DNA that is not their own. And just a little bit. I was going to get rid of this stuff on the board. That's what I was thinking of doing. Okay. All right. Horizontal gene transfer. There's 3 different ways bacteria can do this. Not all bacteria can do every one. Okay. The first type of horizontal gene transfer is called transformation. In transformation, bacteria just take up DNA from the environment. So it doesn't come from another bacteria. They literally grab DNA that's floating around. They take up naked DNA. And again, only a few bacteria can do this. It seems to be kind of random. There's some gram positives and some gram negatives. She's like, can do this naturally. In the lab, we can force most bacteria to be able to do this. But in nature, there's only a few. So again, if you took 161, you transformed E. coli with the P. glow plasmid, that's what you did. But E. coli normally cannot do transformation on its own. Second method is called transduction. And we talked about transduction before. Transduction is when a bacteria gets infected by a bacteriophage, and when that bacteriophage goes to leave, it traps a little bit of bacterial DNA in with the viral DNA, and then it takes that bacterial DNA to the next cell it infects. So, how do I put this? DNA transfer by phage. And every bacteria can be transformed through transduction. This one would be all bacteria. Now, it doesn't mean like you can take E. coli DNA and through transduction give that to Bacillus, because the same phage doesn't infect them both. But one E. coli can be infected, and that DNA can get transferred to another E. coli cell. Right? So you can have transduction within the same species. The third type, the one we'll talk about the most, is called conjugation. And conjugation comes about when two cells literally join, and one of them will give some DNA to the other, usually on a plasmid. Mostly see this with certain gram-negative bacteria. So the DNA is transferred to directly. One bacteria to another by a plasmid. And these are mostly some gram negatives. We're going to talk about plasmids a lot because most of the things that make the really dangerous bacteria dangerous are genes they are either carrying on a plasmid or genes that are transferred through transduction. Right? They're not normal parts of that bacterial chromosome. And so especially antibiotic resistance through the gram negatives tends to spread through conjugation. So we're going to talk about each one separately, and we'll probably get through all three. But transformation is literally bacteria just pulling in DNA from the environment, keeping stuff and seeing if it offers it any benefit. These cells that take up this bacteria are called competent. So take up naked DNA. Competent is the fancy word for a cell that can do this. You know, we can make many organisms work in this manner, but in nature there's only a few. Now, have your bacteria here. It has this chromosome here. You just took up some DNA from the environment. It's normally linear. The bacteria would not want to keep this. Right now, bacterial enzymes are going to see this linear DNA and say, no, 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 and they're going to digest it. So what has to happen is that DNA has to get into either a chromosome or a plasmid to be maintained. So it is very, very rare, but it does happen where it can get inserted, and then it'll be kept and maintained. Linear DNA like this is destroyed. So it has to be part, again, of either the chromosome or it could become part of a plasmid if it's going to hang out and be passed on. And it happens in a manner, if you remember meiosis and crossing over, there's a very similar process in bacteria that will incorporate DNA into chromosomes and plasmids. This is all I want you to know about transformation. Pick up DNA from the environment. Cells that do that are competent. That DNA has to be incorporated into the chromosome or a plasmid. Questions on that one? Okay. That's really it for transformation. Transduction, I already mentioned, this is DNA transfer in a bacteriophage. As we learned with viruses, normally the phage cuts up the bacterial DNA and makes more phage, but sometimes a little bit of that cut up bacterial DNA gets trapped as well. Right. Generalized transduction is random. Again, just some of that bacterial DNA at random gets caught up. Again, to stay within the host, the host has to not die when it gets infected. right? So you have another bacteria here. He just had a phage sit down on him. And the phage is going to inject some DNA. Well, if there's enough phage DNA, it'll destroy this host, right? So that's no good. So this has to be basically an inactive phage because it did not get enough phage DNA. And the bacterial DNA, just like in this example, just like with the transformation, that bacterial DNA, it has to enter either a chromosome or a plasmid to stick around. So that's generalized transduction. Just random DNA is brought. Go back in your memory. Do you remember what a lysogenic phage is? That's a lytic phage. So lytic phages make the cell lyse. What did lysogenic do?
Makes it go dormant.
Why? What does it do with the phage DNA?
It binds it to the DNA, the host DNA.
Yeah, it actually inserts it into the host DNA. So, some phage our lysogenic phage here. Right? It takes its chromosome and puts it in the host DNA. Remember that prophage? That's what so many of our ugly viruses do. They get into our DNA and they hang out. And every time the cell replicates, the virus replicates. Well, when these lysogenic phages decide to reproduce and they pull the phage DNA back out, sometimes they will also pull out some host DNA. So it's not a clean extraction or not a clean cut. Now, lysogenic phages tend to put their chromosome or put their DNA in the same place in the bacterial chromosome every time. So they always pick up the same DNA from the bacteria. Let's see. I don't even have a picture of it. So this is another way that the host DNA or bacterial DNA can be transferred to a new bacteria. Again, this gets trapped in the new phage, and this DNA will go to a new host. I'll step back for a second. I know the microscope's right there. So the only difference between generalized transduction and what's called specialized is specialized takes specific pieces of DNA. Specific bacteria, S-P-E-C, I can spell. Specific bacterial DNA, and this is random. Remember from that early exam, lysogenic conversion, where some of that host DNA will have a phenotype that it'll bring to a new infected cell. I'm trying to go back. So that's a type of horizontal gene transfer. questions on these two? We do the third and weird one. Okay. The third one we call conjugation. Conjugation is literally where two bacterial cells attach to each other. And it is facilitated by something called a conjugative plasmid. Well, usually we call them F plasmids. F plasmid. And what this looks like, you're gonna have two bacterial cells. This one has the F plasmid. He is known as F plus. This one down here does not have an F plasmid. Bless you. It is known as F minus. Hopefully that's straightforward. Right? F plus cells contain the F plasmid. F minus cells do not. One of the genes on this F plasmid encodes what is called a sex pilus. Yes, you can do all kinds of bad jokes later. A pilus is just a long string. So that's what that is. That's created by the F plasmid. And it's like a fishing line. It has a little receptor out here that will attach to F minus cells. So I'm gonna get my eraser. What I'm gonna do with my pilus here, he's gonna, and he's gonna attach to the F minus cell. Right, so this is the sex pilus. And it went fishing, and it hooked to a cell that did not have an F plasmid. Usually they're the same species. Not always 100%. Some of those gram-negative rods, those enterics, will sometimes be a little more, just to keep up with the words, a little more promiscuous. But usually it's the same species. In addition to encoding the sex pilus, they often usually carry other genes, like genes for toxins, or genes for antibiotic resistance, or genes for a capsule, other things that are going to make them really good pathogens. Right. So our sex pilus is connected, and then what it does is it gets shorter and shorter and shorter, bringing the two bacteria together. So this would get shorter, and then it would get even shorter. And when they get really close together, they actually physically connect to each other. And they'll have a small channel between them. Oh my goodness, this is nearly impossible to read here. But the pilus retracts and pulls the donor and recipient cells together, and they make this little channel. Here's where it gets really weird. DNA is double-stranded, right? So if I draw my F plasmid a little bigger as a double-stranded DNA molecule, what's going to happen is this thing's going to be nicked, just one strand. And that strand will enter. So now you have, one strand of the F plasmid here, and you're going to end up with one strand. Of the F plasmid here. So one strand gets transferred to the F minus cell, the other strand stays behind, and then DNA polymerase just makes these double-stranded molecules again. Which I know seems really weird. And then this guy is now F+, and he can go out and infect other cells. Go ahead.
So it, so it injects it into the F negative and then itself again?
So it injects one of the two
Oh,
strands.
that's just an example of how it
And then
should
this-
look after?
Right, so then the
Okay.
second strand will stay behind. So then they'll each just have a single strand of DNA.
Oh, okay, got
It's very weird. It is very, very weird. But this is the most common way that gram-negative bacteria share what we call virulence factors. Things like capsules and toxins and antibiotic resistance. It's extremely efficient, it's extremely effective. It's really the reason people are told to take an entire course of antibiotics. Because it only takes a few of these cells surviving to spread that resistance again throughout all the cells. So you gotta make sure you kill them all. You know, these two are very random and rare. That's very common and directed. Okay. Questions on that one? Because that we'll talk about more, conjugation. All right. Told you this was really short. There wasn't a lot here. But what we're gonna come back to over and over the rest of the semester are these plasmids. So, we're gonna call them mobile genetic elements. Because we can also use transposons, those jumping genes, and we can use phage DNA. There are a few pathogens that are nasty because of phage DNA. But we're especially gonna talk plasmids because they're just very, very common. They're gonna look like mini chromosomes. I don't wanna go over there, I'll be behind that. So a plasmid is a mini chromosome. It's usually, as it says, it doesn't say size, but they're usually like 1,000 to about 20,000 base pairs. 1,000 being the very smallest. So, plasmids. And a mini chromosome. Usually about 1,000 base pairs. They can get bigger than 20,000, but when they get too big, the cell doesn't want to replicate them. It's just too much energy, too much work. So most of the plasmids we see are around 7,000 to 10,000 base pairs. When you think of a bacterial chromosome, and we talked about the chromosome replicating, where did replication start? Just had that exam last week. It was this. Where's this? Here. Anything. Where does replication start? Where does helicase bind and separate the two strands? Heard origin. It's a little bit more than that. The origin is the short term for it. We call it the ORI, or origin of replication. And plasmids will have an ORI. They also often carry something like helicase that recognizes their ORI. Plasmids are semi-specific. Generally, plasmids can work in a gram-negative or a gram-positive. They don't tend to bounce between those two groups. But like I can take an E. coli plasmid and put it in almost every gram-negative you've worked with, and it'll do just fine. So again, very promiscuous. Okay. Can definitely switch species. Sometimes the genus. And in the lab, we can even design them to bounce between gram negatives and gram positives. But that's a design feature. Right, so they're fairly promiscuous. Bacteria want these things because they're gonna carry one or more genes that are beneficial to the cell. So, in 161, okay, we made the bacteria glow green. That's really not so helpful, but you might give it the ability to break down an alternative carbon source. Like when you hear about bacteria that can break down oil. That wasn't natural to them. That gene was put on a plasmid and that plasmid was introduced. Again, when we want to grow things on antibiotics, or the bacteria wants to survive, it'll carry a gene that makes an enzyme that in some way inactivates that antibiotic. Right? So they're going to have one or more genes that are beneficial. So let's say this guy's out in nature, and he's got his origin, and the only gene he is carrying is vancomycin resistance. Okay. So, when is it useful for the bacteria to have this plasmid? Do they need it all the time? No, when do they need it? When you're taking vancomycin or when there's vancomycin present. So the way we get rid of these antibiotic resistance genes in nature is we stop using the antibiotic for a while. Because then none of the bacteria in nature find having this gene useful. And they stop replicating these plasmids and carrying them around. Right? This is how, again, what we're going to talk about for a lot of semesters. This is how this is going to work. They're going to carry around on these plasmids genes that confer benefits. Okay. Don't worry about copy number. Again, most have narrow host range, so most can jump between species. Some can jump between different genera. And as it says here, some can even get into plants. So some of them are extremely jumping around. All right, so what I've been talking about here is what's known as an R plasmid or a resistance plasmid. Those of you going into healthcare are the ones that you're probably most interested in. R plasmids, which stands for resistance, they tend to carry one or more antibiotic resistance genes. And sometimes they carry 5 or 6 different antibiotic resistance genes. So here's an example of one: carbapenicillin, tetracycline, chloramphenicol. And then, of course, you've got to have the gene to make the pillis so you can spread to everyone else in the environment. Right? So these are probably our number one problem when we talk about antibiotic resistance, is these things bouncing around. So if you stop taking antibiotics for not months, but years, you can clear these things from your system. But if you're taking antibiotics all the time, you're just going to encourage the cells to hang on. A little bit more. Ones that you are going to find useful in healthcare. Well, most antibiotics are also included on plasmids. So antibiotic synthesis also comes from a plasmid. I don't think you're going to care about gas vacuoles. Increased virulence, Yersinia, Shigella. So Yersinia is bubonic plague. Shigella causes shigellosis, which is a really nasty foodborne infection. These are both toxins that are being carried by these. Insect toxins, so if you ever wondered about Roundup, not Roundup. I'm getting confused. I'm trying to think what the insecticide is. Raid? Is it Raid? It's not Raid. So they put it in the corn plants, in the soybean plants, so the organisms won't eat them. The GMO plants that everyone hates. So those plants are basically encoded to make the toxin made by Bacillus thuringiensis. And that toxin, if the insects eat it, they die. So that's more of a food one. Yeah, Pilis is toxin production anthrax. So if you didn't know, Bacillus anthracis is in the soil all over, and it's totally harmless. But it has to carry actually 2 plasmids, one for a capsule and one for the toxin. And if it's carrying those 2 plasmids, then it's incredibly dangerous. So, again, most of the things that we think of as making these organisms dangerous come about on plasmids. And those plasmids tend to be these conjugative plasmids that can be passed around quite a bit. All right. That's my happy story for today. That's all that chapter is. Any questions on that? All right. And I'll get up all the chapter 8 stuff too in the next couple days. And we will jump to chapter 14 on Wednesday. So we take a huge jump. I do have your lab exam, or lab lecture exam, so before you go, if you want it, stop by. Otherwise, I have nothing more for you. We're gonna stop there today. If you need to make up any quizzes, today's the day. All right. By the way, please feel free, check my math, because I do add these all up by hand.